about Mendelian genetics. Mendelian genetics is named after, there's a wonderful fellow right here, Gregor Mendel. Gregor Mendel did what everybody else did. At the time, this was in the mid-1800s, everybody did experiments in genetics because they had to optimize the yields from their animals and from their gardens, because you were only one bad harvest away from utter starvation, right, back then. So everybody experimented in genetics. The way they bred their animals, the way they planted, the way they cross-pollinated their plants, always looking for a way to maximize their yields all the time. The difference between what everybody else did and what Gregor did here is that Gregor actually applied some numbers to what his results were. So that's the difference. Everybody did genetics experiments just to optimize their agricultural yields. But Gregor Mendel actually applied numbers to what he saw. And in his kitchen garden, he, of course, was growing peas. Does anybody in here know why May is called the cruelest month? May is called the cruelest month. Do you know why? What? The cruelest. Cruel, hard, horrible. Why would May, beautiful, sunny, gorgeous May, as pretty a month as you could ask for, it was the cruelest month because that was the hungriest month. By the time May rolled around, all of the food that had been generated the fall before was pretty much diminished. So people were going hungry in the month of May. One of the first foods to appear was the garden pea in your kitchen garden. I know that you look at this and you roll your eyes, but if you hadn't eaten a proper meal for at least a month, you'd be jumping up and down for joy, okay? All right, so let's talk about what made Mendel and his friends jumping up and down for joy in May, the cruelest month. So in Mendel's experiments, and the same experiments everybody else did, was he removed the stamens from one pea flower, purple flower in this case, because peas come in different colors. Pink, yellow, white, purple. But in his case, he was working with purple and white. And the stamens are the male part of the plant. The stamens actually have these little pollen bundles on the ends, and pollen is what holds the plant's fur. So what Gregor Mendel did was he did a very controlled pollination. He took pollen from a purple flower and used that pollen from the purple flower to actually pollinate the female part of the plant, which is the carpel. You can do the same thing with a white flower. You can do the same thing with a white flower. So using either a purple flower or a white flower, you can do what is called cross-pollination. This is still done today. If any of you were actually interested in truly doing botany and plant research, this would be a technique that you would use in order to generate new varieties of plants. But in Mendel's case, he had purple flowers, and he took stamens from a white flower and pollinated the purple female. He pollinated the purple female with pollen from a white flower, and of course, the eggs in the carpel would develop into a mature pea pod. What he would do then is he would take these peas, which are actually seeds, and he'd plant them. And in his first generation, after doing his cross-pollination or artificial pollination, took the resulting peas, planted them, and he had a lovely generation of peas. And all of that generation would have purple flowers. What he would notice is he'd say, wait a minute, there are no white flowers. And not only did Mendel notice that, a lot of people would have noticed it. But what he did was he actually took his first filial generation, and that's what F1 stands for. It stands for first filial. And what he did was he cross-pollinated the members of the first filial generation. So the parental generation was the purple flower that had the carpel, the white flower that he got the sperm from the stamen. All of the first filial generation had purple flowers, and what he did was he cross-pollinated, artificially pollinated the F1 generation plants, or hybrids as they're called. They're called hybrids because they come from two different strains. That's why it's called a hybrid. And what he found was that when he cross-pollinated the members of the F1 generation, the resulting garden produced 3 purple-flowered peas, 1 white-flowered pea. So he got the white flower back in the next generation. It reappeared. Okay? If you were doing these experiments, you'd think this was really cool. All right. Not only did he do these experiments with the color of the pea flowers, he did these experiments with the position of the flower, whether it was at the end or middle of the plant. That's called axial versus terminal. He also did these same experiments with the pea color or seed color, yellow versus green, the seed shape. Some of them were round, some yellow, some wrinkled, pod shape, inflated, smooth pod versus a constricted pod color. There are some yellow pea pods versus green, and also stem length. Some of the plants were tall, some of them were dwarf. But what he found, looking at all these different traits, what he found was that in the F2 generation, what he saw was a 3-to-1 ratio. There was a 3-to-1 ratio that came out from all of his breeding. Looking at specific traits, he would get a 3-to-1 ratio. And that's what he got here. The overall numbers are pretty close to 3-to-1. Okay. All right. Is that clear? Are there any questions, or can I repeat anything else? The question is, does the 3-to-1 apply to all of these different traits? Absolutely. And did Mendel look at traits that didn't provide the 3-to-1 ratio? Absolutely. And we'll talk about those later. But it's always best to show your best data. Okay? And that's what Mendel did. It's gorgeous data. There are some experiments that didn't fit, and so what you do is you just sit on them or put them away. And we'll talk about those experiments in our next lecture. Okay. So what I've done is I've summarized some important terms that develop as a result of Mendelian genetics. The first term is allele. Allele is an alternate version of the same gene. So we have 2 alleles for every gene. 1 allele we got from our mother, 1 allele we got from our father. And I'll go into alleles in a little bit more detail in the next slide. And so what he was looking at in these various different experiments was he was looking at alleles. He was looking at the behavior of alleles in his breeding experiments. Because in this case, sorry to keep bouncing back and forth, the purple flower had an allele that encoded purple pigment. The white allele, the white flower, had an allele that was basically mutant. Didn't have wasn't able to produce pigment, and that's why it's white. Okay? So these were the behavior of alleles that he was looking at in his breeding experiments. And what he found with his 3-to-1 ratio was that the two alleles differ, and that one allele can seem to be dominant, where it's going to determine the organism's appearance. Another term for appearance is phenotype. Phenotype can be something that you can see with the naked eye. It can also be something that you can detect using biochemical experiments, and we'll get into more detail about that in the coming lectures. So phenotype is not just what you can see, it's what you can assay for. Not only what you can see, but what you can assay for. Okay? If one allele was dominant, the other allele was considered recessive. It was considered recessive because it didn't have a noticeable effect on the organism's appearance. Okay? Is there a question? Or can I repeat anything? Can I explain anything further? Okay. All right. So what Mendel determined from his first series of breeding experiments where he was getting that wonderful 3-to-1 ratio was he determined what is called the law of segregation. The law of segregation. Because what's happening, if you recall from our lecture on meiosis on Friday, during gamete formation, you have a diploid primary egg cell or a diploid primary sperm cell that are going to undergo meiosis to become a haploid sperm cell or a haploid egg cell. And so during meiosis, what's going to happen is the two alleles that you have for each trait, those alleles are going to separate out into different gametes. And that's why it's called the law of segregation. Those traits, or the alleles that code for those traits that you're looking at, they're going to separate out into different gametes, and that's called the law of segregation. Okay? So now what I want to do is go into a little bit more detail about what is meant by alleles. And again, an allele is an alternate form of the same gene. And so, for example, you would have a allele for the enzyme that generates flower pigment. You would have an allele for an enzyme that generates flower pigment. And in this particular case, and always, alleles are genes. They're genes. They are genes encoding for proteins. And so, for example, if you have an allele that protein was encoding for a purple pigment versus an allele whose protein was mutant and couldn't code for any kind of pigment, and in that case, the allele would be white because there would be no pigment production. Okay? Does that make sense to everybody? And what I want to also point out to you is another term. It's called locus. A locus is the position of a gene somewhere along the length of a chromosome. The locus is the position of a particular gene along the length of the chromosome. But I also want to point out to you, and your book really doesn't go into it, but for every gene you have, you have two alleles. One you got from your mom, one that you got from your dad. Sometimes they are identical, literally nucleotide to nucleotide. But sometimes they are slightly different. They're slightly different. And oftentimes, the changes that you find in a particular, from one allele to the next, oftentimes, if it's a silent change, that means the change is going to happen in the wobble position of the codon of the coding region of the gene, the change is going to happen in the wobble position, where if you have a change from one allele to the next, comparing your mom's to your dad's, it's going to be silent. It's not going to be obvious. Okay? If the change occurs in nucleotide 1 or 2 of a triplet codon, if that nucleotide change occurs in nucleotide 1 or 2, then you're going to have an amino acid difference in the encoded protein, and that can sometimes have an effect or sometimes not. We'll go into that in a lot more detail later on. Okay? Does it make sense to you, knowing what you know about transcription and messenger RNA and triplet codons? If you're going to get any kind of change from one allele to the next, if the change occurs in a nucleotide at the end of a triplet codon, that's the wobble position, and that's going to be a silent change. If the change occurs in nucleotide 1 or 2 of the codon, then that's not going to be a silent change. There's going to be a slight difference in the protein product. Sometimes that can have a significant effect, sometimes not. Okay? All right. Can I explain anything else again, or are we okay? Okay. All right, and so what this does is it demonstrates the law of segregation, where you had Mendel had his purple flowers of his peas, his white flowers, and because all of the members of the first filial generation or the F1 generation were purple, the purple was considered dominant, the white was considered recessive, and we know now the reason why the purple is purple and the white is white is because with the purple flower, the gene that's changed is a protein that is involved in pigment formation, and the gene that is changed in the white flower is a mutation such that pigment can't form. There's always a biochemical basis. There's always a biochemical basis for the difference. But we know just by sheer numbers that the dominant, in this case, is purple, the recessive is white, and when you're doing genetics, the dominant trait is indicated by an uppercase letter. The recessive trait is indicated by a lowercase letter. And so the dominant purple, because there were 2 dominant alleles in this particular strain of flower, there were 2 recessive alleles in this strain of the flower, but the gametes that were produced by each one of these flowers were no pigment or purple pigment, and so the F1 generation was what we would call heterozygous. It was heterozygous because one allele encodes for an enzyme that makes purple pigment, the other allele encodes for a mutant enzyme that doesn't make any pigment. And hence it's called heterozygous. And in the F1 generation, when Mendel did artificial breeding or pollination of the F1 generation, what would happen is these flowers, when they generated their gametes, their eggs and their sperm, half of the sperm had a allele encoding purple pigment, half the sperm had an allele encoding the mutant enzyme, so no pigment. Same thing with the eggs, didn't matter. And so you can do this. It's a fun parlor trick. It's a fun parlor trick, and you're going to do this in lab where you're going to say, okay, let's say the genome, and what we're talking about here is genotype versus phenotype. So the genotype are what we know about the alleles. What are the alleles encoded? So in the purple flowers, the genotype is 2 alleles encoding purple pigment that encode enzymes making purple pigment. We know that in the white flowers, the 2 alleles the two alleles are encoding enzymes that are mutant and don't make pigment. These are both heterozygous. These are both heterozygous. Or I'm sorry, these are each homozygous. This is homozygous dominant, homozygous recessive, and then the resulting generation is heterozygous, one dominant allele, one recessive allele. So that when this generation is making its gametes, whether it's egg or sperm, half the sperm are going to carry the dominant allele, the other half are going to carry the recessive. The same thing with the eggs. Half the eggs are carrying the dominant allele, half the eggs are carrying the recessive. And what you do is you just boil them across. And that tells you, that's going to tell you what the genotype of the offspring are going to be. That tells you the genotype. And from genotype, you can figure phenotype. If you know the genotype, you will know the phenotype. And that not only applies to vegetables in a kitchen garden, it also applies to human genetics. And we will be applying this to human genetics. Okay. But what I'm doing in this first lecture is giving you what we call first principles. What you learn from this lecture is what you're going to be able to apply to all kinds of medical situations. Okay? Does anybody have a question? So the concepts we understand are concepts of allele. We understand the concept of dominant versus recessive. We understand the concept of homozygous and heterozygous. Are you understanding all of these? Can I explain any of them again? Pardon me? The question is, explain the F2 generation again. So these would be our parental generation when you begin doing your experiments. Then, you would breed them together to give you first filial generation. And if your parental generation are both homozygous, then the first filial generation will actually be heterozygous. And then when you breed the members of the first filial generation, that's what gives you your F2 generation. And if you know the genotype of your F1 generation, then you'll know what gametes are being produced. You'll be able to make what is called a Punnett square. You're going to do this in lab. You're going to make a Punnett square, and you're going to be able to calculate how many of each phenotype you're going to get. Okay? Because again, if you know the genotype, you're going to know the phenotype. Question?
What is with the process? How does the cell of the body know which one, whether to translate or transcribe the dominant or the recessive?
The question is, how does the body know whether to translate the dominant or the recessive? And I'm going to go, I've written a very careful study guide for all of you to understand this. All alleles are transcribed and translated, but it depends on the protein product that's being encoded by the allele. So if the allele that encodes the enzyme that's going to make purple pigment, that allele's being transcribed and translated, but the allele the allele here, this allele here, it's also being transcribed and translated, but it's a mutant protein. It's not making any pigment, so it doesn't matter. Always. And that's something a lot of people don't really think about, is that all alleles are going to be transcribed and translated, but it depends on the protein product. And we're going to get into that in a lot more detail, especially when I talk about specific genetic diseases in humans. It's fascinating. Okay. I promise. All right. Okay. So this is where we talk about a little bit, again, phenotype is what you can see by eye or what you can assay for biochemically. Genotype is actually what the alleles are in the organism. So genotype can be homozygous, where you've got two of the exact same alleles, or heterozygous, where the alleles are slightly different. Okay, so these are all basic concepts. Question?
What would you find with that fourth generation, when you just take homozygous to a recessive?
Yeah, so an F4 generation, you just keep breeding down, down, down. Okay? And then, even with the F2, eventually you're still always going to get the 3-to-1 ratio. But again, Mendel always used his best data. All right. Another concept I want to reintroduce to you, if you've forgotten this from high school biology, is a test cross. And a test cross is something that you would do if you didn't know the genotype of your parents. You didn't know the genotype. And when you have a recessive phenotype, that is 2 alleles encoding the mutant protein that aren't generating any pigment at all. You know what the genotype is of this, right? You would know the genotype of, for example, this particular strain of flower because it has no pigment. Therefore, it doesn't have an allele encoding for pigment. No. It has an allele encoding an enzyme that doesn't generate it because it's mutant. So you know the genotype of this one. But would you know the genotype of this guy? It could be homozygous dominant, or it could be heterozygous, where it's carrying the dominant allele and the mutant allele. Okay? So in order to determine the genotype of this fellow, what you can do is a test cross, where you take this strain, you cross it with a known recessive phenotype, because in that way you would know the genotype, and then you look at the resulting ratios in the F1 generation. If this strain of plant is homozygous dominant, and you crossed it with this strain where it's homozygous recessive, all of the sperm or egg from this one are going to have these gametes. All of the sperm or egg from this possible genotype are going to have these gametes, and you're going to get all dominant in the first generation. If, however, you have a heterozygous genotype, what you'll instead get is a 50-50 ratio. Okay? Can you see that? Okay. All right, so the law of segregation. The law of segregation was developed looking at a single trait. The law of segregation was developed looking at a single trait where you were looking at a pair of alleles that encoded a single trait. And these were called monohybrids. They're called monohybrids because, again, you're looking at a pair of alleles that encode a single trait. Okay? However, Mendel also did some experiments where he was looking at 2 different traits and looking at how they segregated out in the subsequent generations. And that led to Mendel's second law of genetics, called the law of independent assortment, where if you're looking at two different pairs of alleles, one pair of alleles is involved in one trait, another pair of alleles is involved in another trait, that what's going to happen is those alleles, those pairs of alleles, are going to segregate independently of each other during gamete formation. And I'm going to go into this in a lot more detail. What I'm doing in this slide is I'm going to go ahead and introduce some other concepts. And the first one is the concept of a dihybrid. A dihybrid means that you're looking at two different traits entirely. So an example of a dihybrid could be seed color versus, for example, seed shape. Seed color versus seed shape. And that's what you're looking at here. A dihybrid is when you're looking at two different traits. And in Mendel's case, he looked at all kinds of different traits. And I showed that on our earlier slide. He was looking at seed color, seed shape, pod color, pod shape, etc., placement of flower, height of plant, etc. And so, for example, in one of his early experiments, he was looking at monohybrid crosses involving seed color and seed shape. And then what he did was he began to follow both traits, the seed color and the seed shape, and watched it over a series of generations. And so things got a little bit more complex. And that's because he was looking at a dihybrid, where he was looking at two traits. And there are lots of different genetic crosses. There are what we call trihybrids, where you're looking at three different traits. When you go up to four traits, things get a little bit unruly. But I've written a very careful study guide to allow you to understand the concepts that I'm talking about today. So let's go to this slide right here. What you're looking at is a dihybrid cross. This dihybrid cross used a parental generation where you had a yellow round, where the pea plants in Mendel's experiments were yellow. They produced seeds that were yellow and round. And the other strain that he was using in this crossbreeding experiment, that strain produced seeds that were green and wrinkled. And what I want to point out again is the dominant allele will determine which letter code you're going to use for that trait. So yellow being dominant over green, the code for yellow is uppercase Y. The code for green is actually lowercase y because it's recessive. Okay? The round shape is dominant over wrinkle, and so therefore the code for round shape is uppercase R. The code for wrinkle is lowercase r. And that's why you're looking at these different letters to describe each condition. All right? Okay. So if these are what we call true breeding. These are true breeding parental plants. You cross-pollinate. The gametes coming from this parent are all going to have an allele that encodes a yellow pea color and a round seed shape. The gametes from this parent are going to all encode green color and wrinkled shape. And when the gametes come together,